Alright, starting Lecture 15, Mitosis and Cancer. This lecture connects cell division, DNA replication, mutations, and cancer. The big idea is, mitosis normally helps the body grow and repair itself, but when cell cycle control fails, uncontrolled mitosis can lead to cancer. The lecture begins by explaining why cells divide. Your body constantly needs new cells for growth, repair, replacing damaged or dead cells. Examples: healing a cut, growing taller, replacing skin cells. New cells must contain the exact same DNA as the original cell. That is why DNA replication happens before cell division. The lecture breaks mitosis into three major ideas. 1. DNA replication. 2. Sister chromatids separate. 3. Cell divides. Mini summary. Mitosis creates identical body cells for growth and repair. Q&A. What is mitosis? Answer: a type of cell division that produces identical body cells. Why is mitosis important? Answer: it allows growth, repair, and replacement of cells. What happens before a cell divides? Answer: DNA is replicated. Why must DNA replicate before mitosis? Answer: so each daughter cell receives a complete set of DNA. What are sister chromatids? Answer: identical copies of a chromosome formed after DNA replication. Next, the lecture explains DNA replication. DNA replication happens during the S phase of interphase. During replication, DNA strands separate, free nucleotides pair with exposed bases, enzymes help build new DNA strands. Remember, A pairs with T, G pairs with C. The slides show that one DNA molecule becomes two identical DNA molecules. After replication, chromosomes contain two sister chromatids connected by a centromere. Mini summary. DNA replication copies genetic information before mitosis begins. Q&A. When does DNA replication happen? Answer: during the S phase of interphase. What does DNA replication produce? Answer: two identical DNA molecules. What enzymes help during DNA replication? Answer: multiple enzymes that copy DNA accurately. What connects sister chromatids? Answer: the centromere. Which DNA bases pair together? Answer: A pairs with T and G pairs with C. Next, the lecture explains the cell cycle. The cell cycle has two major parts: interphase, mitotic phase. Cells spend most of their time in interphase. Interphase includes G1 phase, growth S phase, DNA replication G2 phase, preparation for mitosis. The mitotic phase includes mitosis, division of the nucleus, cytokinesis, division of the cytoplasm. The slides repeatedly emphasize DNA replication happens before mitosis. Mini summary. Cells spend most of their time in interphase where growth and DNA replication occur. Q&A. What are the two major parts of the cell cycle? Answer: interphase and mitotic phase. Which phase takes the most time? Answer: interphase. What happens during interphase? Answer: cell growth and DNA replication. What happens during the mitotic phase? Answer: division of the nucleus and cytoplasm. What is cytokinesis? Answer: division of the cytoplasm. Next, the lecture explains mitosis stages. The professor says you do not need to memorize PMAT terminology, but you should understand the process. Still, the slides show the major stages. Prophase. Chromosomes condense and become visible. Prometaphase. Nuclear envelope breaks apart spindle fibers attached to chromosomes. Metaphase. Chromosomes line up in the middle. Anaphase. Sister chromatids separate. Telophase. Nuclear envelopes reform. Cytokinesis. Cell splits into two daughter cells. The result: two genetically identical diploid daughter cells. Mini summary. Mitosis separates duplicated chromosomes into two identical nuclei. Q&A. What happens during prophase? Answer: chromosomes condense and become visible. What happens during metaphase? Answer: chromosomes line up in the middle of the cell. What happens during anaphase? Answer: sister chromatids separate. What happens during telophase? Answer: nuclear envelopes reform around chromosomes. What does mitosis produce? Answer: two genetically identical diploid daughter cells. Is mitosis involved in making sperm cells? Answer: no, sperm cells are produced by meiosis. The lecture then shifts into cancer. Cancer happens when mitosis becomes uncontrolled. Cancer cells divide excessively, ignore normal growth controls, invade tissues, spread through the body by metastasis. The slides explain that malignant tumors, spread through blood and lymph vessels, continue growing until they damage organs and tissues. Mini summary. Cancer is uncontrolled mitosis that allows abnormal cells to spread and damage tissues. Q&A. What is cancer? Answer: uncontrolled cell division. What is metastasis? Answer: spread of cancer cells to other body areas. Why are malignant tumors dangerous? Answer: they invade tissues and spread through the body. What is a tumor? Answer: a mass of abnormal cells. The lecture then compares benign and malignant tumors. Benign tumors stay localized, do not invade nearby tissue, can still cause problems if pressing on organs. Malignant tumors invade nearby tissue, spread through blood and lymph vessels, are considered cancer. The brain tumor slide shows how even a benign tumor can still be dangerous because it presses on nearby structures. Mini summary. Benign tumors stay localized while malignant tumors spread and invade tissues. Q&A. What is a benign tumor? Answer: a non-cancerous tumor that stays localized. What is a malignant tumor? Answer: a cancerous tumor that spreads and invades tissue. Can benign tumors still be dangerous? Answer: yes, especially if they press on important organs like the brain. Next, the lecture explains cancer cell characteristics. Cancer cells. 1. Lack differentiation. 2. Have abnormal nuclei. 3. Divide without density dependence and form tumors. Density dependence means normal cells stop dividing when crowded. Cancer cells ignore this signal. Mini summary. Cancer cells ignore normal growth controls and continue dividing. Q&A. What does it mean that cancer cells lack differentiation? Answer: they lose specialized cell functions. What is density dependence? Answer: normal cells stop dividing when crowded. Do cancer cells follow density dependence? Answer: no. Why do cancer cells form tumors? Answer: they continue dividing uncontrollably. Next, the lecture explains mutations in cancer genes. The cell cycle has checkpoints that normally control division. Mutations can damage genes controlling these checkpoints. 2 important gene types: proto-oncogenes, normal genes stimulating growth, oncogenes; mutated proto-oncogenes causing excessive growth, tumor suppressor genes, genes slowing or stopping division, correct DNA mistakes. Mutations in tumor suppressor genes can remove growth control. The slides also explain that several mutations usually accumulate before cancer develops. Mini summary. Cancer develops when mutations disrupt normal cell cycle control genes. Q&A. What is a proto-oncogene? Answer: a normal gene helping regulate cell growth. What is an oncogene? Answer: a mutated proto-oncogene causing excessive growth. What is a tumor suppressor gene? Answer: a gene that slows cell division and repairs mistakes. How can mutations cause cancer? Answer: they disrupt normal cell cycle regulation. Do most cancers result from one mutation? Answer: no. Multiple mutations usually accumulate over time. Next, the lecture explains causes of mutations. Mutations may be inherited, result from DNA damage, happen during mitosis. Mutagens, physical or chemical agents causing mutations. Carcinogens, mutagens that increase cancer risk. Examples: UV radiation, tobacco smoke, chemicals, pollutants, viruses like HPV and hepatitis B. The lecture asks, can UV mutations in skin cells be passed to children? Answer: no, because skin cells are somatic cells, not gametes. Mini summary. Mutations can be inherited or caused by environmental mutagens and carcinogens. Q&A. What is a mutagen? Answer: a physical or chemical agent causing DNA mutations. What is a carcinogen? Answer: a mutagen that increases cancer risk. What are examples of carcinogens? Answer: UV radiation, tobacco smoke, chemicals, and some viruses. Can mutations in skin cells be inherited by children? Answer: no. Why can smoking increase cancer risk? Answer: tobacco smoke contains carcinogens that damage DNA. Next, the lecture explains cancer prevention. Ways to lower cancer risk. Avoid tobacco, eat healthy, exercise, protect against UV radiation, get vaccines like HPV and hepatitis B, practice safe sex, get regular checkups. Mini summary. Healthy lifestyle choices and early detection reduce cancer risk. Q&A. What lifestyle choices reduce cancer risk? Answer: avoid smoking, healthy diet, exercise, sunscreen use, and regular checkups. Why are HPV vaccines important? Answer: HPV can increase cervical cancer risk. Why is early detection important? Answer: cancer treatment is more effective when caught early. Finally, the lecture explains why cancer is difficult to cure. Cancer is hard to cure because cancer cells are your own cells. Different cancers behave differently. Tumors change over time. Cancer cells may evade the immune system. Some cancers spread throughout the body. The lecture also explains treatment methods. Surgery removes tumors. Radiation damages cancer cells. Chemotherapy targets rapidly dividing cells. Chemotherapy affects healthy, rapidly dividing cells too, causing side effects like hair loss, digestive problems. Mini summary. Cancer is difficult to cure because tumors evolve, spread, and differ between patients. Q&A. Why is cancer hard to cure? Answer: cancer cells are the body's own cells and tumors differ greatly. What does surgery do in cancer treatment? Answer: removes tumors physically. What does radiation therapy do? Answer: uses radiation to damage cancer cells. What does chemotherapy target? Answer: rapidly dividing cells. Why does chemotherapy cause side effects? Answer: it damages healthy, rapidly dividing cells too. What are examples of chemotherapy side effects? Answer: hair loss and digestive problems.